Hello students, this is Faitan Mirza and we are studying the review for the process of osmosis. Osmosis deals with movement of water. It basically is a diffusion of water, but you never use the term of concentration here. We use the term of water potential. So osmosis, the movement is the ability of the water molecules to move down the water potential gradient from a higher water potential to a lower water potential. Higher water potential is in a dilute solution which is having a less negative water potential, and a low water potential is a concentrated solution which is having a more negative water potential. Pure water with no dissolved solute is said to be having or is given a water potential of 0 kilopascals by convention, and adding a solute lowers the water potential below 0. So it will make the water potential negative. Adding more solute lowers the water potential even further and makes it even more negative. So dilute solutions are less negative, concentrated solutions are more negative. If you apply pressure on the region having a solution, it increases its water potential and brings it closer to 0 and makes the water potential positive. So the application of addition of, you can say, solutes lowers the water potential, and application of pressure increases the water potential. Water potential is denoted by the Greek letter psi, and it is measured in units kilopascals. Osmotic pressure or solute potential is denoted by psi s. It is the ability of the solid particles to pull water molecules towards themselves. The more concentrated the solution is, the more negative it will have the water potential, and more solutes will ensure that it is having a higher solute potential, and they will pull more water molecules towards them. So, for example, there is a solution A and there's a solution B separated by a semi-permeable layer. You can see there's a dilute solution in A and a concentrated solution in B. The dilute solution is having a less negative water potential and a less negative solute potential, whereas the concentrated solution in the region B is having a more negative water potential and more negative solute potential. You can count the number of solutes; they are more in B and less in A. So if it's a tug of war going on between the solid molecules pulling the water molecules, so obviously more water molecule will be pulled towards the side B. So after some time, the solution A will be less in volume and solution B will rise. Why? Because the water has moved down the water potential gradient from higher water potential to low water potential, from dilute solution to the concentrated solution. As already mentioned, osmosis occurs due to difference in the solute potential. Water moves from region A to region B. If you have a semi-permeable layer separating a dilute sodium chloride solution from a concentrated sodium chloride solution, water moves from A to B. As I told you, a high water potential means a less negative water potential, having less solute or less negative solute potential. A low water potential means that it is having more negative water potential and is more negative in solute potential as well. Adding solute to the water lowers the water potential below 0, and the more solute that's added to water, the more negative the water potential be. So keep that in mind. The concentrated solutions have a very low or very negative water potential. And why in osmosis only water molecules move? Because the solid molecules are too large to pass through the semi-permeable layers, the minute gaps that might be present between the phospholipid barrier, which allows the water molecules to move. Keep that in mind that the water is polar molecule. It can still pass through the water through the phospholipid barriers, because the phospholipid barriers is the components are constantly in motion. So tiny openings keep occurring in the phospholipid bilayer, which allow water molecules to move down the water potential gradient from inside to outside or from outside to inside, although water is polar. Plus the transport proteins that allow the channel proteins to function, they are also having the watery pathway. So whatever is crossing through the transport protein, the channel protein, it will allow water molecule to move as well down the water potential gradient. But that process through the channel protein would be fast-like diffusion of water and not osmosis. Osmosis strictly means crossing the phospholipid bilayer. For instance, figure 4.5 shows you there are three cells, one having the water temperature minus 350 kilopascal, the other is having minus 1050 kilopascal, the third having minus 3000 kilopascal. You can see that the water moves from minus 350 to minus 1050 as well as minus 3000. But from minus 1050 cell, it moves to minus 3000 because of the water potential difference. And you can see the more negative the water potential is, the more solute it will be having. In animal cell, the water potential is equal to solute potential. So if you place red blood cell in an isotonic solution, there is no net movement of water. Water moves in and water moves out at the same rate. So the red blood cell stays intact. But if you place the cell in hypotonic solution, water is high outside and water is low inside. The water potential is low inside, so water moves from outside to inside, and as the water enters, the cell swell and the cell burst. If you place the same cell another red blood cell in hypertonic solution, it means that now inside the cell there is a high water potential, and outside the cell there is a low water potential. In this case, water leaves the cell shrinks. When the cell shrinks, it forms spikes. This is called as crenation, and due to crenation, the cell bursts. So animal cell bursts in both cases, whether you place it in hypotonic solution or hypertonic solution, but in isotonic solution, it stays the way it is. When you place a plant cell in a hypertonic solution, water leaves the plant cell and you can see the protoplast is starting to shrink away from the cell wall. This is called the beginning of the plasmolysis. This is called as incipient plasmolysis. You can see that the cell membrane is shrinking away from the cells. The cell wall is a non-living part of the cell, so the protoplast is shrinking away. The gap which forms between the cell wall and the protoplast fills up with the solution which is present outside. So after some time, some complete plasmolysis would have occurred, the cell would become flaccid and the protoplast would have shrunk away from the cell altogether at complete plasmolysis. Why? Because water moved out of the cell, out of the cytoplasm as well as from the vacuole. In plant cells, water potential equals to the sum of solute potential and pressure potential. Now, what is pressure potential? This is what we are discussing. Pressure potential is denoted by psi p. It promotes the movement of water molecules out of that region wherever the pressure is applied. It prevents further entry of water molecules inside. For example, there is a region A and there is a region B. The region A initially had 0.5% sodium chloride and the region B had 0.9% sodium chloride. So you can expect the water to move from hypotonic solution to the hypertonic solution from A to B. But if you have a piston which is present at the top of B, so if you push the piston enough to prevent the entry of water from A to B, water will stop moving. What have you done? You have added pressure from the top. So water entering from A to B cannot come in because there is a pressure at the top. And this pressure is being provided by the piston that you are pushing. If you end up pushing the piston too much, the water might just be driven back into A, and this would be reverse osmosis. This is how you actually purify water in which you take out from C if you want to turn it into drinking water. So reverse osmosis takes place, but anyhow we are discussing osmosis here. So you will apply enough pressure just to stop water from entering. So the red arrows showing you water molecules moving from A to B. You just the cross marks here tell you that enough pressure has been applied that water molecule further cannot enter. So what has happened? What are the effects of increasing solute concentration on the water potential? If you add more solute, the water molecules go away from zero, it becomes more negative. But if you exert pressure, you bring the water potential back towards zero, it becomes more positive or it becomes less negative. This is exactly what the cell wall is doing, and it prevents the cell from bursting. And how so? Where the cell is placed in a hypotonic solution, you know that the cell will start to take up enough water. As this water is entering, the cell will become turgid. The cell will start to exert pressure potential onto the cell, the protoplast. The water potential becomes less negative or positive. Further osmosis prevented, irrespective of the solutes still pulling water molecules inward, the cell becomes turgid. So there will be still be various solute molecules present inside the cell, which is now placed in a hypotonic solution, and they will still be pulling water inward. But since the cell wall is there, it starts to behave like a piston. It starts to exert the pressure on the protoplast, preventing further entry of water. Because if that further entry of water would have occurred, the cell would have kept on swelling and it would have burst the way the animal cell burst. But the presence of the cell wall prevents this from happening. When the protoplast is turgid, when it has taken enough water and it cannot expand beyond that, the cell wall starts to exert pressure all around the protoplast in the same way as the piston was applying in the region B. This is called as the pressure potential. It prevents further entry of water irrespective of still how many solute molecules are there inside the cell pulling water from outside the hypotonic solution. So further entry of water is prevented and hence the cell will not burst. That's it for now.